Good morning.
Morning, everyone. Thank you for being on time. We're going to go ahead and get started. So we have quite a packed week today. We have to finish up lecture 3, and hopefully we can finish lecture 4 as well. And that would help us set the groundwork for doing a lab next week. The lab, like I said, it's going to be done virtually online. You do not have to come to campus. I'll tell you more about it um, later on. But first, let's do a quick recap. Has been, 6 days since we last met, and I think it's a good idea to just reorient ourselves and see where we are at. So we were talking about the endomembrane system, which includes the endoplasmic reticulum, right, the rough and the smooth, the nuclear envelope, the Golgi body, as well as something called the lysosome. Right? So the rough ER is the endoplasmic reticulum that has ribosomes on them. Okay? So ribosomes are made in the nucleolus, the center of the nucleus, and they are these tiny dots that actually has a large and a small subunit. The ribosome themselves are made up of proteins as well as something called rRNA, ribosomal RNA. Okay? So some of these ribosomes will be floating on its own in the cytoplasm. Others will be attached to the endoplasmic reticulum. The ribosomes and the endoplasmic reticulum can work together to produce and package proteins. Right? Once you created a protein, it will pinch off from the rough ER and form a vesicle. The vesicle will then migrate to the Golgi body right here, right? Also known as the Golgi apparatus. And the Golgi body is responsible for modifying the contents inside the vesicle. Right? It will sort them, it will tag them, right? Add different things to it, remove certain things, and then it will repackage it into a new vesicle. This vesicle can be released from the cell for immediate use. It might stay in the cell until it is needed, or it could become part of the cell membrane. And sometimes it could even become the lysosome, which is another part of the endomembrane system. Now, the lysosome is basically a specialized vesicle that has a mixture of enzymes in them. And these enzymes are responsible for breaking down various things that enters the cell. Maybe a cell has engulfed some food particles. Maybe if we are talking about white blood cells, it would be engulfing some kind of bacterium. So whatever comes into the cell that needs to be broken down, it will go to the lysosome, and the lysosome will use the enzymes that's in it to basically break down the bacteria, break down the food particle, and so on and so forth. Sometimes you would have organelles that are worn out in your cells, and you kind of want to recycle them. You want to break them down and unlock the nutrients that are locked in them, maybe reuse some of the resources, and that's when lysosome is going to come in and do its job as well. So it can break down things that are coming into the cell, break down worn out organelles or macromolecules that are already inside the cell. And lastly, we talked about how lysosome can trigger apoptosis as well, right? That is programmed cell death. When a cell gets too old or when it's not performing its function properly, you want to destroy the cell. Okay, so these are deliberate um, you're killing off cells deliberately, purposefully. Okay, and that's where we left off. Does anybody have any questions before I move on to the next part? All right. So the only thing that's left to talk about in the endomembrane system is the smooth endoplasmic reticulum. It's a continuous part of the rough ER. The only difference between the smooth and the rough ER is well, functionally they're different, but then appearance-wise, the difference is whether or not there are ribosomes attached onto them, right? So let's go back to the PowerPoint slides here. And this is the slide about smooth ER. The function of smooth ER is to produce lipids. Okay, rough ER produces proteins. Smooth ER produces lipids. Right. So there are various kinds of lipids that we learn, right? Phospholipids, triglyceride, steroids. These are all examples of lipids, and you will make them in the smooth ER. Um, for example, we talked about steroid hormones, right? The testosterone, the progesterone. So you would expect cells in the ovaries or testes to have more smooth ER. Okay, so one thing that you should be aware of is that not every cells are going to have the same number of each organelles. Right? It depends on the function of the cell. They might have more of one kind versus the other. If we're talking about the cells in the ovaries or in the testes, right? Their job is to make the sex hormones, right? The testosterone, the progesterone. So in those cells, you would expect to have more smooth ER. Okay? In another cell that has another job, right? Then you would expect to find more of another kind of organelles. Okay, so they're not like really fixed in ratio. It really depends on their function. Other jobs of the SER, the smooth ER, is to break down fat as well as detoxification of drugs. So if you're taking medication, right, antibiotics, that kind of stuff, and they need to be metabolized, the smooth ER is going to play a role in that. Another interesting thing is the cell can increase or decrease the number of organelles to match the current demand of the body. Right? So for example, if the liver is exposed to drugs, so like let's say you're on antibiotics, right? Okay, so all of a sudden there's a demand to break them down, then it's possible for the liver cells to increase the amount of SER in it, right? To help cope with this sudden increase in medicine in your body. Okay, and when you finish your course of drugs, then you don't need as many SER, and then you can kind of lower them again. So it's not static. Okay, the situation it's dynamic, it's fluid. It's going to match the demands of the body. Okay, so again, rough ER is for synthesis of proteins. Smooth ER is for synthesis of lipids, as well as breaking down lipids and fats and detoxification of drugs. We talked about all these things already, and that's it. Okay, so ribosomes are made up of. Now, for the interest of time, I'm just going to tell you the answer. We'll do more polls later. Ribosomes are made up of rRNA as well as protein. Okay, so B and C. Which of the following organelles are involved in breaking down macromolecules, toxins, and waste? That would be the lysosome as well as the smooth ER, right? So let's move on and talk about other organelles in the body, in the cell. The stuff we're going to talk about now are no longer part of the endomembrane system. Okay? So there is something called peroxisome. Peroxisome. They look very similar to a lysosome. Like if you look at a real picture of it, like that's a peroxisome here and that's a lysosome. They are just two different circles with different shades, right? It's really hard to tell. So I will never ask you to label these things on a picture, right? It's like you need to be trained to distinguish them. But in any case, the inside of the peroxisome contains chemicals that would allow it to perform oxidation reactions. We will learn more about oxidation reactions later, but oxidation basically means removing electrons. Okay, so these are reactions that are that will involve taking electrons from certain molecules, and in doing so, you will break them down. Okay, one of the byproducts of these oxidation reaction is H2O2. H2O2, you might know, is hydrogen peroxide. Okay, hydrogen peroxide, you can buy them from drugstore. It's a disinfectant, right? For your wounds, for example. It comes in a brown bottle usually. But your body, the peroxisome, they produce the hydrogen peroxide as a byproduct of their reactions. And it turns out H2O2 are very toxic for the cell, so they must contain it within the peroxisome. And then what kind of things do they break down? Well, macromolecules in general. Okay, so now there is an additional place to break things down, right? We know the lysosome breaks things down. We know the smooth ER break things down. The peroxisome also break things down. Okay, the difference is how they break it down, right? With the lysosome, they use a mixture of enzymes. With the peroxisomes, they use oxidation reaction. Right? So again, it plays a role in detoxifying poison, such as alcohol in the liver. All right. So a lot of things are kind of overlapping. Right? They don't really um, work at things alone. Right? It's more like redundancy and kind of working together um, as a whole. Right. So that's peroxism. It produces peroxide, right, as a byproduct. Okay, let's see if we can do this question. I'll let you read it first, and then I'll launch the poll. All right, it's almost 1 minute. I only have 5 people who are answering, unfortunately. Okay, that's fine. So the correct answer is C. Okay, the picture that we drew, right? Look here. The vesicle can be released immediately. It can become part of the cell membrane. It can stay in the cytoplasm, or it can become a lysosome, right? And the peroxisome is not part of the endomembrane system, right? So uh, that's why the protein has no business doing anything with the peroxisome. All right. Next, we're going to move on to mitochondria. Now, I want to draw this mitochondria with you to help you understand it better. I think this is your note. Is this your maybe this one? I don't remember. One of them. They all. I teach the same class 3 times each week, so sometimes I get confused as to which note belongs to which class. They're all the same thing. Okay, like it doesn't really matter. So let's draw a mitochondria together. All right. Okay. Singular is mitochondrion. And then if you change it to RIA, then it's going to be plural. So this is the powerhouse okay, of the cell. Powerhouse of the cell. It's the site of cellular respiration. Okay. You might remember cellular respiration from lecture 2 when we talk about the function of carbohydrates, right? The cellular respiration equation goes like this. We have glucose, and we're going to react it with oxygen to produce carbon dioxide plus water plus energy. Okay. Now, there are many different kinds of energy, right? You can have like chemical energy, electrical energy, solar energy, wind energy, so on and so forth, right? But the usable form of energy in the body is something called ATP. Okay? We'll talk more about that later. Okay? That's the currency of energy in a cell. Right? So part of the cellular respiration takes place in the mitochondria. And that's the equation. That's the chemical process. All right. Now let's draw this mitochondria together. We have a big oval here that represents the outer membrane. Okay. And then we're gonna have something like this on the inside. Right? So that's the inner membrane. Okay, so outer membrane is kind of smooth. Okay, I'll slow down a little bit. Outer membrane is smooth. And on the inside, the inner membrane, it's folded up. It's folded up into these kind of like an accordion, right? We call them finger-like projection. Let me see if I have a paper here. Right, so like this is the inner membrane, right? Rather than smooth, it folds up like this. It folds up into these structures. All right, so by folding up, it actually allows you to pack more membrane, right? So the inner membrane folds into a structure called cristae. Which maximizes surface area. Okay, we're trying to do the cellular respiration reaction, right? So the more surface that's available, the more reactions can happen at the same time. Okay. Now, in terms of the outer membrane and the inner membrane, it's the same structure as the cell membrane. Okay, it's also made up of phospholipids, you know, with a similar structure, you know, with some cholesterol in it, perhaps some proteins, so on and so forth. Any questions? You guys understand what I'm trying to do here? Like think of this as a bag inside a bag. There are two layers, right? A Ziploc bag, put another Ziploc bag inside. So now you have a membrane within the membrane, right? Any questions? So the space that's between the outer membrane and the inner membrane, that's called the intermembrane space. understanding the structure of the mitochondria is going to be important for lecture 5. When we learn the cellular respiration process, we need to know the different parts of the mitochondria so that we know what goes where. Okay. And then on the inside of the mitochondria, the innermost portion, that's called the matrix. Okay, that's called the matrix. So in the different parts of the mitochondria, you would have different parts of the reaction happening, and ultimately it's going to generate ATP. Okay, so it's going to produce ATP. Right? And ATP can be used by the cells to do various things. Okay, to move things around, to do chemical reactions, to build things, all that require energy in the form of ATP. Okay? All right. So let's go back to the slide here. That's what it looks like, the real thing. All these lines are the foldings, the cristae that I was telling you about. I'm not going to read this to you. It's basically the same thing I was telling you about. Okay, the fluid inside the inner membrane is called the matrix. The finger-like projection called the cristae. Okay, you should read it yourself. But again, it's what I told you. The main function, again, right? It's that the mitochondria is the powerhouse. Okay, it produces ATP for the cell. All right. There is another structure called the chloroplast. The chloroplast. Okay. Chloroplast is found in plant cells, not in animal cells. Okay. Uh, can also like I mean it's found in plant cells, not in animal cells, but also in some we some other cells. Okay, some other cells. They are called protists. I mean, you don't really have to know the name, but what you do have to know is that there are more different organisms out there besides animals and plants, right? Okay, there are other categories. Some bacteria, for example, right? There are the amoeba and the paramecium that I showed you. So they belong to other kingdoms, we call them. Okay. So if it's just animals and plants, then chloroplast is found in plants, but not animals. But if you include other kingdoms that we haven't learned about, some of them also have chloroplast. The chloroplast contains chlorophyll that allows the plant to carry out photosynthesis. Okay, so chlorophyll is green. That is what makes the plant green, and it's a chemical that allows the plant to capture sunlight. Okay, and sunlight allows the plant to do photosynthesis. All right. Now, a lot of people have this misconception. They think photosynthesis makes energy for the plant, but that's not true. Photosynthesis makes food for the plant. Okay, so photosynthesis makes glucose for the plant. Right? And glucose is food, right? And it's actually the opposite of cellular respiration. So it takes carbon dioxide. It takes carbon dioxide plus water, and then with the help of sunlight, it will make oxygen and glucose. Now that glucose is going to be stored as food for the plant, right? Okay, in the form of starch. Then the plant can use that in cellular respiration. Okay, the difference between us and plant is plant has to make their own food. We eat our own food, right? And then you will use it to burn it, to burn the glucose to produce energy. Okay. Chloroplast is also double membrane, just like the mitochondria. Okay, and then there are these stacked fluid-filled membrane called thylakoids. Don't worry about the last one. I'm not going to ask you about it. Okay, just erase that. Never mind that. Okay, so in this picture, you can see chlorophyll. Some people buy chlorophyll, right, online. My student was telling me that they buy from like Amazon or something, and then it comes in a concentrated bottle, and then they add it to their smoothies or something. You can get chlorophyll naturally just from like eating vegetables, right? Like the darker it is, the more chlorophyll it has. So like spinach, kale, they have a lot of chlorophyll. There's no health benefit, not that I know of consuming, pure extracted chlorophyll. Okay, like there's no nutritional value to it, right? Like, but it's like a gimmick, right? As with most health product. So that's Chloroplast. Are you guys okay? So by now, right? Like you're pretty deep into the program already, and you have had a couple of tests and quizzes, and things are starting to pile up, right? And this is where the time management is really important. Don't let things pile up too much. Your next quiz is not until after Thanksgiving. So you will have some time to prepare. It is on lecture 3 and 4. I will give you a survey after class, a link. I'll post it on eCentennial, and then you can give me some feedback. It's anonymous. You will not I will not know who gives me the comments. Okay, so please feel free to speak your mind. Madison asked, when do we get our midterm marks? I mean, um, I don't know how to answer that. Hold on, give me a second. Now you guys take math, right? So you should know how to calculate your marks. But you should be able to see your marks on. Is that correct? Am I correct that you can see your marks on eCentennial? Like you saw your quiz grade, right? Is that correct? Can someone confirm that for me? I posted already. Have you guys seen your quiz grade? Okay, so
Yeah.
that's your mark, right? Now, so you will be able to see that as you do more quizzes, do more assignments for me, right? But for some reason, nobody knows why, okay? Like the total grade of all the stuff doesn't show up. I can't get it working, okay? For some reason, I don't know why. So you have to calculate that on your own, right? So I've created this thing for you. If you go to useful information, there is something called grade calculator. Okay, so you can download the grade calculator, and then you can plug in the stuff that you plug in your grades, basically. Let me see if I can download it to show you. Yeah, but like you're not going to use your midterm grades to apply, right, Madison? Like you need to have completed the program before you apply, right? And I believe like you're not going to get, we don't have midterm grades that are official, right? You will only get your final grade at the end of the course, right? So over here, if I open the grade calculator, let's see if it's gonna come. There we go. All right, so right now you have to enable editing, of course. Right now there's nothing, okay? But you have your quiz 1 grade. I don't know what you get. Maybe you got 75, so you plug that in, right? So this is your current grade over here. This is your current grade. And then You can also key in the final grade that you want. Let's say you want a 70 in the final grade for the course, then it tells you what you need to get in the remaining assessments. Do you understand? So your current grade is 75. If you want a 70 in the end, you will need to get 69, an average of 69 on everything else in the program, in the course. Okay, so like quiz 2, quiz 3, quiz 4, so on and so forth. Everything else needs to be 69. Do you guys understand? Right? So like if you get quiz 2, right, later on you get 80, right? And then your test 1 is like 40. Sorry, let's do better than that. I don't know, like 67, something like that. Right? So as you do more evaluations, your total grade will change, right? And then you can key in what you want in the end, so that you can have an idea of how well you need to do for the remaining assessments. Okay? So that's how you can calculate it. I don't know, but that's a really long answer for what you have asked. Are we together? Any quick comments about the quiz? I mean, you guys did okay, reasonably similar to all the classes. The average was pretty standard. But any quick comments about the quiz? Was there any surprises or anything like that? Yeah, it was all right. Okay. Well, anyway, like I said, right? Like if there's something you really want to tell me, you can do it through the survey. Survey only has 3 questions. It will ask you, what you want me to continue doing. So things that you like and you want me to do more of. What you want me to stop doing, as well as things that are that I'm not doing, and you would like to see. I felt it was worded very differently than how we were taught. Yeah, that's a common thing. You got to understand, right? Like, I am not the only one who's teaching the course, right? We have 3 different teacher teaching the course, and we all contributed to the test questions, right? There are literally thousands of questions in the bank, right? So that's why sometimes, like, if the word seems a little bit different than the ones I've used, maybe someone else wrote the question. And it's not unfair or anything, because for the other prof, when they see my question, right, it would be probably a little bit different than how their teacher phrased it as well, right? Okay. But don't focus too much on how it's phrased. The content should be things that we've done. Okay. All right. So again, right, speak your mind freely in the survey, okay? So that I can know what you guys think truly. But keep it professional, right? Like don't like be angry at me or anything. You can voice your frustration, but keep it professional. Now this is an important slide, okay? Important slide. All right. This shows you the relationship between chloroplast and cellular respiration. Okay, you should write down the things that I'm saying, or you can watch this again later. Okay, this would be at the 30-minute mark of the recording. All right, and again, this is important slide. All right, you can imagine why that is the case. So mitochondria are found in both animal and plant cells, right? Whereas the chloroplast are only found in plant cells and not in animal cells. But the two are related, because for the reaction that takes place in mitochondria, cellular respiration, the ingredients, the reactants, are the products of photosynthesis. Right? Let me write it down for you. For cellular respiration, right, it is C6H12O6 glucose plus oxygen, and it produces carbon dioxide and water. For photosynthesis, it's the other way around. It's CO2 plus H2O produces C6H12O6 plus O2, right? So you can see they are actually complementing each other, right? The product of one reaction becomes the reactants of the next, right? And that's how we cycle carbon, essentially, right? In the ecosystem. All right. So the difference is for plants, they make their own glucose and then they burn it in their mitochondria. For us, we have to eat the glucose, right? We can eat the glucose from the plants, right? From the starch that they produce, and then we burn it in the mitochondria. Okay, so the key thing to know here is that the two processes are complementary to each other, right? The reactants of one are the products of the other, okay, and vice versa. Let's talk about other things that are found in a plant cell. We have this thing called central vacuole. Central vacuoles are these big structures that are taking up a lot of volume inside the cell. They are there to store water and nutrients, and sometimes they would have central vacuole in the colored part of a plant, okay, like the flowers. And in those places, the central vacuole will be responsible for storing pigments. If the plant is poisonous, they might also be able to store poison in the central vacuole. This is a paramecium. You saw it earlier in one of the slides. It's not an animal cell. It's not a plant cell. It's considered as a protist. Okay. Like I said, right, there are different kingdoms, right? Animal kingdom, plant kingdom. There is a kingdom called Protista, okay, which is made up of protist cells, and they live in pond water, fresh water. And one of the challenges of being a single cell, a unicellular organism in an open water, is that water will constantly come in, okay, and fill up the cell. And if you don't remove those water, it will get too big and burst, right? So they have this thing called a contractile vacuole. Okay, it's basically a pump, right? So it will squeeze and it will pump the excess water out of the cell to prevent it from bursting. Okay. Plant cells, of course, also have cell walls, right? It's not the first time we learn about cell wall. They are made of cellulose. They provide structural support, prevents the cell from bursting. Unlike the cell membrane, the cell wall is not selectively permeable. It doesn't regulate things, right? Just pass right through with no problem. Okay. Let's take a look at this one. Select all the false statement. Read it first. I'll launch the poll. Okay, that's it. Select all the false statement. So smooth ER is part of the endomembrane system. That is correct. It's not false. There is an abundance of mitochondria in muscle cell. That makes sense, right? Mitochondria is needed for making energy, and muscle cell has a lot of movement, right? You need constant supply of energy, so you would expect to have more of those. All cells have cell wall to protect it. That's not true. We don't have cell walls. Hydrogen peroxides are found in peroxisome. Peroxisome, not lysosome. And chlorophylls are found in chloroplasts. So the correct answers are C and D. Those are the false statements. Now we're going to move on to talk about cytoskeleton. Right? Cytoskeleton. Cytoskeleton are basically these structures, usually they're very long, that are found inside the cell, and they are there to provide support for the cell shape, for the structures, as well as to guide movements of different things. So there are three types. They are listed here. Microfilaments, microtubules, and intermediate filaments. Okay. Let me just quickly show you what each one does. This one is for, you know what, actually, let's just go through the slides. It's probably easier that way. Okay, so three types of fibers, okay? Microfilaments, the intermediate filaments, and the microtubules. Microfilaments is made up of a protein called actin. Right? Each of these circle represent an actin, and then you just end up forming a very long strand. And you take two of those strands and you twist it up together, you get your microfilament. Right? It's found throughout the entire cell. This here is a cell. Now, you can't really see the cell membrane, but that's where it is. Okay, this is kind of like the cell membrane surrounding the cell. That's the nucleus. And the person who prepared this slide, they stain the actin protein. You actually can stain different parts of the cell using different antibodies, okay? So they tag the actin here, and then it has a little fluorochromes that gives off green colors. In any case, you can see the actin is essentially everywhere in the cytoplasm, okay? So they provide attachment sites for the different organelles, right? You know, the mitochondria is not just floating around on its own randomly, right? Think of this as a spider web, right? And then you can stick the mitochondria on it, right? Stick another mitochondria in it. That endoplasmic reticulum, right? It sticks it right there. So it provides a scaffold, if you would, to attach different organelles onto it. Okay. So that's one function of the microfilament of the actin. The other function is a little bit harder to visualize, okay? Some cells need to move. So let's say we have a cell here, okay? And then the cell wants to move this way for whatever reason, okay? Cells need to go catch bacteria or something like that, so they need to move around. So here are all the actins, right? Here are all the actins in the cell. And what happens is if the cell needs to move, it would break down the actin. Oh my goodness. It would break down the actin from the back and shift them towards one side, right? So that allows this part to kind of extend forward, right? So, you know, if I'm a cell like this and I want to go that way, like I would push all the actin out and then form an arm, and then I'll grab onto the surface and like pull myself up, right? I don't know if you can visualize that. That was a really weird performance. But you know, afterwards, it's basically like this. The cell would extend and grab on, okay, and then push more actin forward and then drag the rest of it forward. Do you understand that? right? Think it's just like this, and it's just crawling around, okay, crawling forward. Okay, every time it needs to extend, it needs to push the actin to one side. That's basically how the actin works. All right, so provide attachment sites for the organelles, and it allows movements. Okay, that's it for microfilaments. Next, we have the intermediate filaments. Think of them as staplers, okay, or tapes. All right, they stabilized structures. Sometimes we want to lock two adjacent cells together. There are parts of the body where you need the cells. So like that's one cell, that's another cell, and you want to staple them together so nothing leaks through between the cells. Give me an example of a part of the body where you don't want things to be leaking out. Yeah, exactly, right? Skin, right? Like, you put some lotion on, right? You don't want the lotion to leak all the way into your bloodstream, right? So, you have some of these intermediate filaments to keep a tight seal. Okay, these tight seals are called intercellular junctions. And any other place, honestly, I wasn't expecting skin to be the answer, but that's a good example. Anywhere else? Like inside your body, is there somewhere you don't want things to be leaking all over the place? Yeah, the stomach, right, Rory, right? The intestines, exactly, right? You got some food in there. You don't want it to spill all over your abdomen, right? So you need to keep them there, right? Blood vessels, you don't want your blood to be oozing all over the place. So you need to keep a tight seal between the cells. And that's what the intermediate filaments are for, okay? They are staplers, they tape things together, right? Now, inside the cell, we want to have a pretty stable structure for the nuclear envelope as well, right? Okay, because we don't want the DNA to be leaking out and stuff like that. So there's a special kind of intermediate filament that supports the nuclear envelope. We call them the lamins. All right. There is a condition that is associated with lemon instability. Right? Because it's a protein, that means if the instruction to make it is changed, right, from the DNA, you're not going to have the right kind of protein being made, right? And so people who have inherited a form of lemon a mutation causes their nucleus to become unstable, okay? And it also affects the chromatin structure and gene expression, and that results in a condition called progeria. Right? You might have seen pictures like these on the internet before, right? Where the person looks really old, but then there's something childish about the appearance, right? And that condition is called progeria. This person is actually about 14 years old, even though he has all the features of an elderly person, right? Thinning of the hair, right? The wrinkles and all that stuff. So it turns out the mutation in lemon A causes accelerated aging. Okay, and there are no cure for progeria, and these people die, relatively young. Have you guys seen this before? Maybe some of you. Yeah. Okay. If not, you can look it up. There was actually a movie that was made about it. This was a long time ago. Some of you probably weren't even born, but I'm sure it's on YouTube. But they got it. They try to depict this scenario, but then they didn't quite get it right. Okay, like in the movie, the person just grows up to be like an adult really, really early on. Like, after 4 or 5 years, they become like a teenager. But that's not true. Okay, like you're still very young. It's just that the body is aging. Okay. Wonder if there could be a reverse of this. Yep. So there are some research that involves gene editing, right? We're going to learn about gene editing later on in lecture 9. But because it's a DNA condition, right? A mutation. So if you theoretically, if you change the mistake, you can rescue the situation. Now, damage that are already done, you cannot undo it. But if you identify this early, you can prevent the accelerated aging, right? And there has been some promise in mouse model. Okay, in mouse model, we have yet to see success in humans. But it's, you know what, you start doing the experiment in mice, but a lot of them, like some 90% of the studies that works in mice is not going to translate well into the human version. Okay, so it's still a long way to go. All right, so we had the intermediate filaments, which are what we just talked about, and then we have the microfilaments, which is the scaffold, right? The green stuff that I showed you to attach the organelles on to help the cell move forward. And then there is the last one, which is called a microtubule. Okay, you know that vesicle that butts off from the rough ER, and then it's supposed to go to the Golgi body, right? Like, how does it know where the Golgi body is? The cell is pretty big relative to the size of the vesicle. Like, how does it know where to go, right? Like, if we just rely on random motion, diffusion, for example, like it might never get there, right? So that's where the microtubules come in. Microtubules are these, like, you can think of them as like conveyor belts inside the cell. Right? So the vesicle can hop onto one of these conveyor belt, and it will travel along the microtubule and get to its destination. Okay, I kid you not. There are these molecules that looks like a person without the head. And then it would carry the vesicle on these arms, and then it will walk along the microtubules, so literally carrying the vesicle and then walk along and then bring it to, yeah, like a stickman without the head though, okay? It will carry the vesicle to where it needs to go, right? So it'll be taking on the vesicle from the rough ER, and then it will bring it to the Golgi body along these tracks. These are for real, like you can actually see them under the microscope. Okay, amazing stuff that's happening. In addition to that, they also provide structure support. What you're looking at here is the formation of a new connection in a neuron. Okay, you might have remember seeing some neuron cells earlier. I showed you, and they make these. They have these long extension that comes out, right? That forms like a connection with another neuron, right? We call them synapse, right? So like if you need to grow a new one, then the microtubules will help guide the growth of these extensions. Now there is an other organelle called centrioles. Centrioles look like this, and it's found in animal cells, but not plant cells. Okay, I'll have a chart coming up that summarizes what's in plants only and what's in animals only. But the centrioles are important because they produce a special kind of microtubules called spindle fibers. Spindle fibers. Remember, I told you during cell division, we have to move the genetic material around, right? We have to move the chromosome around, right? So something has to guide the chromosome movement, right? Again, nothing is random. If you wait for things to happen randomly, it will take forever, okay? So we need to have some kind of a guiding system. And that's what the spindle fibers will be doing. Right? It will move the chromosomes to the intended location. Okay, so centrioles will send out these specialized microtubules called spindle fibers. You can also find microtubules in things like cilia or flagellum. Usually cilia covers the entire cell. Okay? So like the paramecium I showed you, it has cilia all around it and it helps it swims around. The cilia is made out of microtubules. In the human body, right, the airway has cilia. Right, so this one will help move trap dust particles, pollutants out of the system. Okay, and over here we have the sperm. And sperm has flagellum. Okay, so cilia covers the entire cell. Flagellum, usually there's one. I mean, there are some protists, again, in a different kingdom. They may have like 2 flagella. Okay, that's possible. But for humans, the sperm only has 1 tail. Right? If a sperm has 2 tails, it would just swim around in circles. You can find anything in YouTube, on YouTube. Like, just do it after class or something. Type in like sperm with 2 tails, and then you will find videos of a sperm with 2 tails swimming around in circles. Okay, we don't want that, right? We want a single tail to help it move forward, and that's made up of microtubules. This is the fallopian tube, right? That's the tube that connects the ovaries to the uterus. And when the ovary releases an egg, right, the cilia will help guide it to the uterus. Okay. So here is a list of things that are found in the animal cells only. So lysosome, centrioles, and flagella. And these are things that are only found in plants. Chloroplast for photosynthesis, central vacuole for storing water, nutrients, sometimes pigment and poison, and then cell wall to provide structural support and prevents it from bursting, right? Okay, which of the following is made up of 2 intertwined actin protein strands? Oh, you guys are not really participating this morning. That's okay. I would just end the poll so we don't waste too much time. The correct answer is the microfilaments, right? The 2 red ones, right? That I showed you earlier. And that's it. That's it for lecture 3. Now lots of things going on in the study guide that you guys need to go through. Okay, on quiz number 2, in quiz number 2, I will show you some of these pictures. I will show you a picture of that maybe, and then you will have to tell me the names. Right, some mitochondria, right? I'll show you a picture of this. You will have to tell me is centriole. Right, so you don't know which picture I'm going to show you, but you will have to recognize them. Okay. There are like a lot of good questions to do here. I'll post the answer after class, of course. And then there is a diagram for you to work on as well. Now, I said this before. I don't know if you guys heard me the first time. When you see the study guide, not only will there be the Word document, right, with the answers in them, there's also a video that you can watch. These are older recordings of me going through the study guides with the class. Okay, so you can use those as well. It's not uploaded yet for lecture 3. I will do it after class. Okay, 1 and 2 are available for now. All right, let's take a break. When we return, we're gonna do lecture 4. If you haven't already downloaded that, please get a copy. We will resume at 9:35. All right, see you in a bit. All right, let's keep going. Hey, I just have a quick question for you guys. Does the drawing help or not really helping? Okay, let me ask you to do a poll for me. Like all the drawings that we're doing, if you think it's helpful, please choose A. If you think, not really for you, please choose B. Should I draw? I'm gonna draw more if you guys like that. Yeah, you guys like it?
I like words better.
You like what better, sorry?
I like words.
Words better? Yeah, no drawings. Okay. All right, thank you. Majority of you seems to like it. So I'm gonna keep going with the drawings. Okay. So this is lecture 4. Lecture 4 is about transport, right? How things move in and how things are leaving the cell. And of course, controlling what comes in, what comes out. That's the job of the cell membrane, right? Which is why we're going to be looking at the structure of the cell membrane. And that is called the fluid mosaic model. Okay, for those of you who are planning ahead, like how big should your page be and so on and so forth, this is what we're drawing. Okay, this whole mess is what we're drawing. Okay, so plan your space accordingly. So this is a cell, and we're basically taking a piece of the cell and then looking at it. Okay, so let's draw some phospholipids. Now, when you draw the phospholipid, I want you to deliberately put a few that has a kink in it. Okay, you can put like, I don't know, like 3 or 4 or something like that. Okay, just include some with kinks in it. Okay, so this here is the phospholipid bilayer. Right? It has 2 layers. That's why it's called bilayer. It's made up of phospholipids. And you should know by now that this is the hydrophilic head, and then sandwiched in between are the hydrophobic tails, right? Now, this is the outside of the cell. The outside of the cell is called ECF. Okay, it's outside. ECF stands for extracellular fluid. I'll type that in the chat box. Extracellular fluid, ECF. Okay, I just don't have space to put in here. So that's the outside. The inside of the cell is, of course, the cytoplasm. Okay. ECF is mostly water. Cytoplasm is mostly water as well, which makes sense to have the hydrophilic heads oriented that way. Okay. So for the phospholipid bilayer, the percent of saturated and unsaturated fatty acid depends on the cell type. Okay, you want to understand this for a particular question on the lab. I will explain more later. So far, so good? I hope so. Now, we're gonna draw more phospholipids here. I'm gonna leave a gap here, okay? Draw more phospholipids. You can have some more like kinky ones here with the double bond. Okay, I'm gonna leave another gap. And then just leave another gap. These are phospholipids, okay? The ones that have the polar head, the glycerol, and the 2 fatty acids. Now in the gaps, I'm gonna draw some proteins. That's what these purple ovals are. Let's make it thicker. Okay. Now, some proteins will go all the way through. Others will just be like, attaching on the side. Right? Think of this is a loose analogy. Okay, it might help, it might not. Think of like a bread, a piece of bread, like a raisin bread. Okay, some raisins will go all the way through the bread, right? You can see it on both sides, right? Some raisin would just be, stuck on one side, did not go all the way through. That's what these proteins are like. Right? So the ones that are like all the way through, we call them integral proteins. And then we have some that are just, on one side. They are the peripheral proteins. Okay, and they have different functions, which of course I will love to tell you about them in the upcoming half hour. Some proteins will have sugar on them. And that's what these green little lines are, the little hexagons and whatnot, glucose molecules. And that makes them a glycoprotein. Glyco means sugar. A glycoprotein here. It is protein with carbohydrates. And again, I will tell you what they do in a bit. And then you will have some that has lipids on them. Okay, lipids. Like lipids will do the fatty acid like that. Okay. And these would be called lipoproteins. Protein with lipids. Okay. So there is another peripheral protein, if you want to label it. It can be on the outside leaf or the inside leaflet. Okay. Now, I'm not going to write this next part down. You can write it anywhere on your page if you have it, okay? But listen carefully. Listen carefully. This is called the fluid mosaic model. Fluid means it's not static. If you think about like the wall behind me, that's not fluid. It's solid. Nothing is moving on the wall, right? But this is a fluid mosaic model, meaning, listen carefully, please, meaning this phospholipid is not stuck in place. That phospholipid is free to move left and right. Okay, the best way to visualize this, and again, like it's hard to, like, I imagine you guys don't have this in real life experience. I never seen it before either. But like when I think of this, I think of filling up a bathtub, okay, and then we pour some balls in it, okay? You know those balls that kids play? Like my kids have them when they're younger, right? Those plastic balls, right? You just pour them all into the bathtub, right? And then you imagine all those balls just, moving around, right? It's not static, it's not fixed in place, right? The ball can move past each other easily, right? So those balls would be kind of like the head, the hydrophilic head of the phospholipids. It's able to shift from one place to another. Do you understand? Okay, so that's what fluid means. It's able to move laterally, and sometimes, okay, very rarely, they would be able to flip to the other side. Okay, so lateral movement is common. Flipping is rare. Okay, so phospholipid I'll write some down. Phospholipids can move laterally. Okay. And sometimes, only sometimes, flip to the other leaf. To the other side, we can say. Remember, there are two leaves, right? Two layers. To the other layer, we'll say the other layer. Any question? Now, some of you who are wondering, why are we learning this? Okay, why do I have to know this? Know that the pre-health program is ministry standardized. Okay, like you go to another college, you will learn the same thing, right? It's not me, right? I'm just following the guidelines, so don't shoot the messenger. All right. Okay, so sometimes people get disgruntled, like, oh, he makes us learn all these like extra things. It's not me. I'm just following the guidelines. Okay. All right. What else is on this membrane of ours? Well, we are gonna pop some cholesterol in there. Okay, so there's some cholesterol right here. You're gonna put another cholesterol here. You can draw like 4 carbon rings if you want. Okay, so these little stuff here, those are the cholesterol. Right? Cholesterol helps maintain membrane fluidity. Okay. In general, more cholesterol equals more fluid. Okay. And as you will learn in a bit, depending on the environment that the cell is in, sometimes you want it to be more fluid, sometimes you want it to be more solid. And on the inside, in the cytoplasm, you would have these lines. Okay, we learned about these earlier before the break. Anybody want to tell me what those might be? What are those blue lines, huh? Matrix. The matrix is like the inside of the mitochondria, right? That's where we used it in that context. That's right, the actin. Very good, the actin. Right? I'm gonna call it the cytoskeleton, right? Cytoskeleton, the actin. Right? So it helps maintain the cell shape, right? And so on and so forth. Are we together on this? Any question about fluid mosaic model? Mosaic just means it's not one thing. You know, in chemistry you learn about homogeneous, right? A solution is homogeneous when everything is appearing the same phase, just one uniform phase. Mosaic is the opposite of that. Like a pizza is mosaic. You can see the different parts to it, different toppings, right? So the cell membrane is mosaic because it has different parts to it.
Question.
Yes, Georgie.
I know you probably asked this already, but I was having trouble logging on. The beetroot lab.
Yes, I haven't talked about the beetroot lab. Okay, we will.
Oh, good.
Yeah.
I didn't
So
miss anything.
we will do that. I might talk about it. Probably not this week. Okay, I'll talk about it on Tuesday's class, and then you will work on it on Tuesday, and then you will submit it on Thursday. Okay.
And sir, question again.
Mhm.
If like me, if I feel like we did bad on this first test,
Mhm.
are we gonna get another chance to make it up at any time? Please say yes. I'd like probably
So we
reading
talked
week.
about this before, right?
I know.
Again,
I
it's
figured you did.
just right. So it's not just me. As the program,
Oh,
we have a policy where we do not allow rewrite of a test, right? And this is the reason is to be fair for everybody, right? You know, if a
It don't even
rewrite,
have to be a rewrite. It just could be a makeup test, if you know.
same thing, right? Same thing, right?
Oh.
Because now certain individuals have an opportunity for extra grades that others don't, then it's not going to be fair, right? You know, sometimes later on in the course, for example, quiz number 4 is really hard. Okay, people usually do very bad on that one for some reason because it's a lot of genetic problem solving. So for that one, I will give you 2 chances. Okay, you will have 2 attempts, and then I will only count the best one or something like that. Okay,
For
so
which one?
for quiz number 4, which is you don't have to worry about it now because that doesn't happen until like way later in the course. Okay, so
Yeah.
there are situations like that, but generally, if someone does not do well, we don't have extra, like you cannot do an extra assignment for extra grades. That's not something that we offer. Okay, I know it's not ideal. It's like disappointing answer, but it's a program policy. Okay. All right, so that is that. Question about the model before we move on. Okay. Let's go back to here. So lecture 4, membrane transport. A lot of the stuff we talked about already, right? We basically drew this thing. Okay, and again, like I personally believe drawing it out is much better for learning than for me to show you a picture and just talk about it. But now that you've drawn it, you can study this diagram, compare this with what we've drawn, right? And see if it makes sense to you, right? So there is the integral protein that we talked about, and then there's the peripheral protein. There's some cholesterol. We talk about glycoprotein, okay? Sometimes you can even have glycolipids, right? Even though I didn't talk about glycolipids, but you know glyco means sugar. So these are lipids with carbohydrates on them, right? That tells you the different components of it. Okay, we have the phospholipids acting as the major ingredient. We have the cholesterol, which helps maintain the correct fluidity. I wrote it down for you already. More cholesterol means more fluid. The sugary protein and the sugary lipids, they play a role in what's called cell identity. I'll elaborate on that later on. It's coming up, don't worry. This is an important slide. Important slide. Okay? So there are 2 important slides, right? Okay, the one that in lecture 3 and this one. You know what? I'm just gonna tell you. Okay, these 2 slides are gonna be the type out questions, right? The written response question for your next quiz. All right. I told you guys what was gonna be on the quiz, like last time, like the written questions, and still it wasn't done as well as I hoped it to be, right? Some of you didn't even answer. Some of you left it blank. I
Sir,
don't
that
know why.
question was, I find that I thought it was gonna be, more. I didn't have time to answer that question, be honest.
You didn't have time, but you ran out of time.
No, you
Oh.
have to think about that question and really answer it in detail, but I
Yeah.
didn't find out. Boy, I don't know.
Anyone else find time to be an issue?
I had no problem with it.
You have no problem with the time?
Yeah,
Okay.
I have a problem with the time.
You have a problem with the time? Yeah. Okay.
Understandable.
Yeah, yeah. Okay. I mean, like, these are the things you can tell me, okay, in the survey. All right. Be honest. Okay. I'll see to it. If a majority of you is having problem with time, that's something I'm flexible with. Okay. But like, I mean, like, I cannot give you like 2 hours, right? That's not realistic, right? Okay. But I can extend a little bit. All right.
I didn't even check my mark, and I know I failed that test.
No, you should check your marks. The marks are up, right? It's important to check your marks so that you know where you stand, right? Yeah, do not check. Anyway, my point is this, guys. All right, the next quiz, you will also have 2 written response question, right? Okay, what's the first question? Anybody remember? Please tell me. What's the other one that I starred? Earlier today, I put a star on another question that I said is very important, right? What slide was that? Anybody remember? Oh boy.
I don't remember. I tried to block that out of my memory.
You deliberately set up a mental block, right? Okay, and thank you. Okay, let me say it one more time. Yeah, let me say it one more time. Okay, for your good, for your own good, right? Okay, someone asked, can we get a quiz one answer? I like, as I said before, right? I cannot share the quiz answer with you because we need to maintain the integrity of the test bank. And I know you are like, okay, then how can we study for the test, right? Don't worry, like I'll pick out some questions later on to discuss with you guys. Okay, so this is the slide. This is the important slide that I was telling you about. Okay, what number is this? This is number 48. Number 48. Okay, slide number 48. Okay, one of the long answer questions, the written response question on quiz number 2 will be based on this. Okay, if you forgot everything I said on this slide, when you watch this recording, it's at the 30-minute mark. Okay, 30-minute mark, 30, write it down. Now here is another one. This is another important slide that you have to know. This is the other short answer question that you have to do on quiz number 2. And if you want to watch this, this would be at the I guess, what is it, like hour and a half mark of the recording. Okay, 1.5 roughly there. So this is the question. How would the percent of saturated, unsaturated fat and cholesterol content of a cell membrane found in plants growing in cold environment differ from those growing in hot environment? Okay. So what do you think? Or a plant that is growing in
Sunlight?
cold environment. Okay, so someone mentioned something about sunlight, but let me finish first. This plant is growing in cold environment. What you're trying to tell me is, do you think there is more or less saturated fat in the phospholipid? Okay, do you think there is more or less unsaturated fat? Do you think there is more or less cholesterol? Okay. All right. Like, some of you are saying more, some of you are saying less. I have no idea which one you guys are referring to. Is it like more, or more, less? Like, what is it, right? I'm gonna give you a minute to think about that, and then I will tell you the answer. All right. Remember, I'm looking for 3 things here. For a plant that is growing in a cold environment, how will these things change? By the way, some of you missed the first quiz for whatever reason. Okay, please send me an email. Right, tell me why you missed the quiz, and then we can set up something. Okay, you don't need to give me a note. Right, the first time I let you go free. Okay, you just send me an email, and when you send me an email, please tell me your name. Okay, a lot of you like to send me 1 or 2 lines, and you don't tell me your name. I can help you. Okay, tell me which section you're from. You're from 201. Okay, so in a cold environment, right? Listen to me carefully. In a cold environment, is the cell membrane more solid or more liquid? If it's cold, do you expect things to be more solid or more liquid? Think of butter, okay? If it's cold, is it more solid or more liquid? More solid.
More solid.
Exactly. That's the first thing you have to understand. Cold environment means cell membrane is more solid. Okay. Nathan, maybe stay behind after class to talk to me about the situation. Okay. So that is more solid. Now, we don't want the cell membrane to be completely solid, because if it is, nothing goes through, right? Which is why the modifications that are needed is going to make it more liquid, right? To counteract the cold environment. Let me say that one more time. The cold environment makes it more solid. I need to change the cell membrane to make it less solid, right? In a cold environment. So how do I make it less solid? Do you want more saturated fat or less saturated fat when you want to make it? Okay, so we need to make it less solid or more fluid, okay? More fluid. So we need less saturated fat, right? Because we learn saturated fat is solid at room temperature. So I need less saturated fat to make it more fluid. How about unsaturated fat? Do we want more unsaturated fat or less unsaturated fat if we want it to be more fluid? Okay. More fluid means more unsaturated fat. That's right. So the percentage of unsaturated fat is going to go up, right? Think back to our drawing. That means you will have more phospholipids with a kink in it, right? So that way it could be more fluid. What about cholesterol? Do we want more cholesterol or less cholesterol? What do you think? That's right, also more, right? To make it more fluid. So these are the adaptation you would expect to see in a plant that is growing in a cold environment. Okay, let me just go through the hot environment scenario with you. If it's really hot, think of a plant that's living in like the desert or something. The high temperature will make the membrane very liquidy, right? Very fluid. We don't want a membrane that's too fluid. Everything is just going to leak down. So the adaptation will need to make the membrane more solid. And in that case, you would need more saturated fat, less unsaturated fat, and less cholesterol. Okay? So think that through yourself. Right? Don't just memorize it. Okay? You have to understand it in order to answer the question. So Ramon, you asked, how does the plant survive the cold environment? You mean? Yeah, so like these adaptation allows the membrane to have the right consistency, right? So that it will continue to basically function, even though it's at a cold temperature. Okay, I'm not sure if that's what you're asking or okay, all right, great. So that's that. Okay, now don't say I did not tell you. This is gonna be on the quiz. Now we're gonna spend the last 15 minutes talking about membrane protein functions. We know there are integral proteins that goes all the way through. We know there are peripheral proteins that are just either on the outside or on the inside. But what are the proteins for, right? What do they do? There are 6 functions that we're gonna look at, and that will be the end of today's lesson. Okay, so just to refresh your memory, the green one is the integral, spans all the way through. And then we have the peripheral protein that are either attached directly on the phospholipids, or sometimes they are like a buddy to the integral protein. They attach to the protein instead of the lipids. First function. First function is attachment to cytoskeleton. You know this picture we drew, right? We have all these cytoskeleton here, right? The blue one. That is the actin. So they have to stick to something, and that's what the proteins are there for. Right? The cytoskeleton will actually basically attach to these proteins. Right? Think about laying a tarp on the floor, like at a park or something. Right? Like if the wind comes, it's just gonna flap all over the place, right? So you gotta anchor them in place, right? With those pegs. So the proteins are the anchor. Right? We don't want the cytoskeleton to detach from the cell membrane. We need to attach them. So that's what one function of the of the proteins are. They're there to provide attachment sites to the cytoskeleton. Allows the cell to attach to the outside as well, to the extracellular matrix. right, which is the material that's outside of the cell. By providing anchorage points for the actins and sometimes the tapes, right, the intermediate filaments, it allows the cell to maintain a particular cell shape. As you already know, red blood cells, they look like this with a very distinct indentation in the middle, right? So how do you maintain that indentation? You need to have the cytoskeleton attaching to the membrane proteins. Okay, so these are all the fibers that are outside of the cell. If the cell wants to attach to them, they also need to have the membrane proteins. So that's the first function. Second function is cell signaling. All right. We already talked about hormones a couple of times. There are protein hormones, there are steroid hormones, and hormones in general is responsible for changing how a cell behaves. Right? It's a signaling molecule, right? It gets released from one part of the body, and then it travels to another part, and then it causes the cell to behave differently. Right? So here, these are like we can call them like we just say hormones. Okay, it will bind to a integral protein, and then the protein will transmit the signal inside the cell, which then causes the cell to do various things. Here is an example. There is a molecule here, epinephrine. Some of you might be more familiar with the other name, which is adrenaline. So let's say a person is getting ready for a race or something, right? You do the warm up, you get yourself, all pumped up, you get some adrenaline in your system. So one of the function of adrenaline is to increase glucose availability, right? So how does it do it? Well, it will bind to a receptor. The receptor is a membrane protein, and then it will cause all these things that are happening inside the cell, right? We call this a signal transduction pathway. It's a really complicated name, but all that means it's just a series of reactions. Okay, and then it will ultimately cause hydrolysis of glycogen, and then you will release the glucose. Okay, so the molecule never comes into the cell, right? It just binds to a receptor on the cell, and then it causes all these commotion inside the cell, all these reactions inside the cell. Whenever I talk about this example, it reminds me of my childhood. Okay, my family does not like Halloween. I never found out why. Okay, but every Halloween, my mom would be like really anxious. Okay, so anytime someone rings the doorbell, she'd be like, turn off the light, hide in the basement. Okay, pretend no one's home. I never really understood that. I mean, if you turn off the light when someone rings the bell, like they know you're home, right? Like they know you're just ignoring them, right? Okay. But this kind of like it's like what's going on in the cell. Okay. This protein is like the doorbell. The kids are like ringing the bell. They never got into the house. They never got into the cell. But all these actions are going on as a result of the hormone, binding to the receptor protein. Okay. So it's able to relay certain action from the hormone to all these things that are happening inside the cell. Okay, the kid never came in, but everybody inside the house was scrambling. Okay, the hormone never came in, but all these reactions are happening to change the behavior of the cell. Okay, so first function is attachment to cytoskeleton. Second function is cell signaling. Here is another example of cell signaling. You don't have to know this one. I just showing you foreshadowing. Next semester, we'll learn about how neurons communicate with each other, and they will release these signaling molecules. It will bind to a receptor and then cause the neuron to behave differently. You might have heard of things like dopamine, right? Serotonin, right? You might have heard of Prozac, which is an antidepressant drug. They all work with this kind of cell signaling mechanism. Next, we have enzymatic activity. Right? Enzymatic activity. You know enzymes. Right? Enzymes are proteins. And sometimes it's easier if you put proteins that are related to each other next to each other. Okay, what do I mean by that? Here is a reaction. Okay, let's say you have starting material A, and then enzyme number 1 will change it to B, but it's not done yet. Okay, you got to do another reaction that change it to C, and you need enzyme number 2, and then you need to change it to D, and you need enzyme number 3. So enzyme 1, 2, 3 are related to each other because they belong to the same reaction pathways. So rather than having enzyme 1, 2, 3 randomly scattered in the cytoplasm, it makes sense to put the enzyme right next to each other, right? So when the initial substrate, the initial ingredient comes in, you can immediately produce the next one and then feed it to the next enzyme, which will create the next one and next one. So you can have reactions happen rapidly, one after the other. Okay, one after the other. And this increased efficiency, it increases the speed of which you can complete a reaction. Okay, so by putting enzymes that are related to each other, close to each other on the membrane, you allow reactions to happen quickly. Number 4, intercellular junction. Right? Earlier today, I told you the intermediate filaments are like staples to glue cells together, right? 2 cells next to each other, you want to have a tight seal. It turns out those are not the only thing that allows cells to stick together, right? Membrane proteins will also help you reinforce the connection. Okay, just having the staplers are not enough. You need to have, extra things. Maybe put some duct tape there as well, right? To glue it together. Okay, so the intermediate filaments, they are like the staplers. The membrane proteins, in this case, they are like the duct tape, right? So extra things that glue them together. Make sure the cells are extra tight. Nothing leaks through. Okay, it's scary if you have some of these junctions breaking apart. The disease that are associated with it it's like straight out of a horror movie. Okay, like you would have literally parts that are falling off inside the body, and then things start to leak all over the place, right? That could lead to internal bleeding, hemorrhages, and it's just not a pleasant situation. Okay, next function of membrane protein is cell-cell recognition. Remember those glycoproteins and glycolipids that we drew, the sugary proteins, right, and the sugary lipids that we have, like these ones, right? Collectively, they make up an identification tag for your cells, okay? Everybody has unique identifiers on their cells. Okay, your cells are going to look different than my cells. Okay, no one will have the same set of identification tag as you unless you have an identical twin. Okay, even your kids will have similar identification tag as you on their cells, but not identical ones. Okay, that's why you can't just take anybody's liver and then transplant it into someone who needs it. Like, it's gonna get rejected, right? Okay, that's why you have to match the donor and the recipient. And even when you try to match it, you cannot have 100% match because of these ID tags. These glycoproteins, these glycolipids create a unique identifier for your cells. And anytime your immune system detects a cell that has different ID tags, they will attack it. Okay, so when a bacteria goes into your system, it has a different set of ID tags. Your immune system would attack it. Right, so this here is a white cell, and then it's eating, whatever the heck this is. I don't know what it is, but it has different kind of ID tags. That's how it knows, and it's gonna, get rid of it. So it's a way for your body to distinguish self versus non-self. All right.
I have a quick question.
Sure, go ahead.
So you said the big yellow one was a white blood cell, was
Yeah.
it?
That's right.
The white blood cells are not white.
They are not whites. Yeah, they are only whites
Oh.
because again, like you can color them, right? When you look at them, you know, you can actually color them any color you want. Like they just chose to have yellows, right? We can,
Okay.
yeah, these are called pseudo color, right? It's depending on the person who basically photoshopped the picture. They're like, you know what, let's make them yellow today. That's basically what it is. Okay. So anybody know your blood type, by the way? Blood type, anybody? We'll share them. Okay, so some of you see B positive, two B positive, excellent.
Be positive.
B positive, okay, looks like it's that's the winning ticket of the day. B
My
positive.
doctor did not inform me of that after I'd taken many blood tests.
Really? Oh, that's a bummer. If you donate blood, that they would also tell you in some cases. Okay, O positive, great. Okay, so what does that have to do with what we're talking about? Well, it turns out if you are A, then you have glycoprotein A on your red cells. If you are B, you have glycoprotein B on your red cells. Okay, it gives you again like cell identity, right? And next semester we'll learn about transfusion compatibility, right? Like if you're type AB, who can you give blood to, right? Okay. Justina, is there anybody O negative here, right? O negative is sometimes they call them universal donor, right? But you know, I have an issue with that name because it's not true. Just because you're O negative doesn't mean you can donate to everybody. And you know, we're gonna find out why next semester. I am also AB positive. Excellent. It's good to know there's someone else in the class that shares the same blood type. Okay. So if you're AB, right, it means you have the glycoprotein A and the glycoprotein B. That's my point, okay? They create these identities for our cells. All right, last one, and then I'm gonna let you go. Last function is, I knew someone with RH blood type. Actually, you know, when people say they are positive or they're negative, the positive and the negative is referring to the RH. Okay, that's basically what it means. If you are positive, it means you have the RH. And if you are negative, it means you don't have the RH. Okay, RH is named after the monkey called like rhesus monkey. And since we're kind of related to monkeys, you know, we share some blood type with them. Anyway, last function of membrane protein is transport. Okay, which is of course the topic of today's lecture, right? The title of today's lecture. So in order for you to move things in and out of the cell, right, sometimes you would need to use the help of the membrane protein. Okay, so that's it. 6 functions. You should review them yourself and you know, fill it in the study guide. I have 2 questions here, and then we will end. According to the fluid mosaic model of cell membranes, which of the following is a true statement about membrane phospholipids? I'm just going to go through it with you because of time. First one, they are free to depart from membrane and dissolves in the surrounding solution. That is not true. What that is saying is one of these phospholipids can just, you know what, I don't want to be part of this membrane anymore. Let's leave, right? And just pops out like that. That of course is not true, right? Like if that's the case, then you might lose your whole membrane, right? So that's why it's false. They occur in an uninterrupted bilayer with membrane proteins restricted to the surface of, of the surface of the membrane. That's not true. We have proteins on either side, right? And sometimes proteins go right through. Next one, they have hydrophilic tails in the interior of the cell membrane. That's not true. The tails are hydrophobic. They can move laterally along the plane of the membrane. That's true. Remember my analogy with the balls and the bathtub, right? They flow around laterally, no problem. So that's the correct answer. Last one, they frequently flip-flop from one side of the membrane to the other, right? Not frequently, they rarely, right? I told you, rarely. So the answer is D. Okay. Which of the following is not a function of membrane protein? Okay, so I'm just gonna quickly recap it for you. Attachment to cytoskeleton, right? Cell signaling, cell recognition, enzymatic activity, transport, and also I forgot what the other one is, but anybody remember which one I missed? Let's just go back and see quickly. And oh, intercellular junction, the staples, right? Okay, so these are the six of them. And over here, the one that I did not mention would be protection from lysis. Lysis is to break the cell, right? Okay, so that would be the function of like a cell wall or something. Okay, that's not the function of membrane proteins. All right, well, thank you for coming to class again. As you can see, we are losing 1/3 of the class already. So, good job for coming here. I will post the survey link shortly after class, as well as the recordings and everything. Please take a moment to do that. I will see you all on Thursday, okay? Enjoy the rest of your day. Goodbye.
Thank you, sir.